answer one question I always hear from folks at events, like, yes, this event will be recorded, and you will be able to access the recording during the event and after because it's live streamed. So we're just getting that set up. Julie says hello from upstate New York. Hello, Julie. All right, and I'm going to put the YouTube link in the chat just so everybody has a record of it. Oh my gosh, people are coming on in. Hey, everybody, introduce yourself in the chat. Tell us where you're calling in from. If you want to put your zip code in there, that helps us keep track of who we're reaching and where. So great to hear from you all. All right, and we're right at the 6 o'clock Eastern mark. So I'm not going to delay. I'm going to go ahead and get started. Let me share my screen. So hello, everybody. My name's Caroline Nickerson, and I'm from the SciStarter team. I'm so excited to be joining you all tonight in partnership with the Network of the National Library of Medicine, the All of Us Research Program, to talk about today's featured project, the eterna project, with our featured speaker, Joo-Soon Kim. We're going to tell you all about citizen science and how you can get involved in real, meaningful scientific research from wherever you are in the world with any background and any level of experience. We're playing with a different format today. So today we're going to do our normal presentation, our normal live stream, up until there's about 20 minutes left in the event. Then we're going to switch to breakout rooms, and you'll be able to participate in the project yourself. And the breakout rooms will not be recorded. So our recording and our live stream will stop about 20 minutes before the event is over. This event will last 1 hour total. Until we get to our breakout rooms, the event is closed captioned and also has American Sign Language interpretation. If you need any help accessing that, feel free to chime in the chat. We are here to help you. And you'll also have time to ask some questions in the breakout rooms as well. So without further ado, I'm going to get us started with some polls. Well, I want you all to introduce yourselves. Tell us where you're calling in from. If you want to put your zip code in, that's great too. If there's something you're really excited to learn about in relation to eterna, you can say that as well. If you're particularly fascinated by RNA-based medicines, let us know. I'm looking at the chat and seeing who's introduced themselves so far. We have Mike from Butler, New Jersey. Margaret from Tucson, Arizona. We have Michael from Antarctica. You know what? I believe you, Michael, because there is citizen science in Antarctica. We have someone from Minnesota, California. Oh my gosh, what a great group. Greenwood, Indiana. Someone from Alaska, Connecticut. All right, welcome everybody. Thanks for being here. Keep introducing yourselves. We're so excited that you're interested in learning about citizen science and how you can turn your curiosity into real world impact. So we want to know a little bit about you. Have you ever participated in a citizen science project before? There are no wrong answers. If you're not sure, that's okay. We'll give you everything you need to know to participate in the eterna project. If you have participated in a project, let us know which project it is in the comments in the chat. We'd love to know that. You know, if you're watching us on YouTube, you can chime in the YouTube comments as well, even if you're watching this recording after the fact. We just want to know a little bit more about you. So get those answers in, make your voice heard, vote. I'll give you a few more seconds to vote. 3, 2, 1, and I'm going to end the poll. Let's share those results. So it looks like about 65% of you have participated in citizen science before, and quite a few of you, 35%, have not. Both are totally fine. You are in the right place to do citizen science today. All right, let's go to our next slide. Now we want to know, have you heard of the All of Us Research Program before? They're our partners for this event. So I always like to ask this question to find out who's in the know and who might not know. Get those answers in, everybody. And while you get those answers in, it looks like folks have done the Stall Catchers project before, really cool. And folks have done the Audubon project. So it looks like we have some birders in the room with us today, which is great. Okay, get your answers in. I'm going to give you a few more seconds to vote. 3, 2, 1, and I'm going to end the poll. And now I'll share my results. So it looks like about 57% of you have heard of the All of Us Research Program before, and about 43% of you have not. And for those of you who haven't, You're going to hear from Tess Wilson today about how you can get involved with all of us. And for those of you who have, hopefully you'll get a helpful refresher about why this program is so important. So I'll stop sharing those results. And now we have our last poll question. We want to know who you are. Are you an aspiring citizen scientist? Are you a parent of an aspiring citizen scientist? Are you a teacher, an educator, a troop leader, etc.? Are you a librarian or part of a library staff? Are you a researcher or a project leader? Are you just a bored person on the internet? Or are you none of these? All of these answers are totally fine. All right, get your votes in. Looks like we have quite a few aspiring citizen scientists in the room with us, quite a few educators as well, some library staff members, some researchers. We have a few bored people on the internet. I'll give you all a few more seconds to vote. So 3, 2, 1, and we're gonna end the poll. And we'll share the results. So it looks like about 22% of you are aspiring citizen scientists. About 9% of you are parents. We have 39% educators, 26% librarians or library staff, 9% researchers, 4% of you are bored people on the internet, and 17% of you identify with none of these categories. Well, that's totally okay, because by the end of today, all of you will feel like citizen scientists. So we'll stop sharing the results and we'll move on. So you've been hearing this word citizen science a few times so far, but you might be thinking, what is it? Citizen science is a collaboration between scientists and researchers and those of us who are curious, concerned, and motivated to make a difference. Basically, citizen science is any time you volunteer to move science forward. And this can be by collecting data, analyzing data. It just means that you're a meaningful part of the scientific process. And the reason we're spotlighting Eterna today is it's such a valuable project. Just by solving puzzles, you can make a difference. You can help contribute to different discoveries in the medical field. And citizen science is as diverse as science itself. It's everything from health to nature to outer space and everything in between. So no matter what you're curious about, there's a citizen science project out there that you can do and you can meaningfully be a part of. And a great way to get started is by making a SciStarter account. At SciStarter, our goal is to connect you to the projects you can do. So we allow you not only to discover new projects, but also to track your participation. There are over 3,000 projects, events, and tools listed on SciStarter. And we're getting closer and closer to our biggest event of the year, Citizen Science Month, which is in April. So you can pretty much find something to do every day on SciStarter, but a really good place to get started is scistarter.org/NLM. So I'd urge all of you to go ahead and go to that URL. Feel free to minimize me and type that into your chat to the browser window and make your SciStarter account, because that's your passport to all these different projects. You can join them, keep track of them, discover new ones. You'll be part of an ecosystem of citizen science in no time. Plus, it's a lot of fun. Also on the scistarter.org/NLM page is the Introduction to Citizen Science tutorial. Anyone can do it. It was designed by SciStarter, the network of the National Library of Medicine, and Arizona State University to introduce people to the what, the how, the who, and the why of citizen science. And it's offered in English and in Spanish. This tutorial is meant to be accessible to different levels. You can use it in your classroom. You can just do it as an adult if you want a refresher on what citizen science is. There are quiz questions. It's a lot of fun. It's interactive. There are videos. And you also get a certificate at the end. So I saw quite a few of you are educators. You could use this tutorial with your community. If you're a library staff member, you could use the tutorial in your online programs. There's really something for everybody. So feel free to go ahead and click that link in the chat to get started. And at this point in the presentation, before we get to Doosan Kim and the Eterna project, I'm going to pass the mic to Tess Wilson from the network of the National Library of Medicine to briefly tell you about the All of Us research program. Tess?
Hi, thanks, Caroline. So my name is Tess Wilson. I'm the Community Engagement Coordinator for the network of the National Library of Medicine Middle Atlantic region. And today's webinar is brought to you in part by NNLM, the Network of the National Library of Medicine, which is a program of the National Library of Medicine and works together with the All of Us research program. So I'll talk to you a bit about All of Us today. It's a research program of the National Institutes of Health, the NIH, and it's seeking 1 million or more people across the US to help speed up medical research. People who join will share information about their health, habits, and what it's like where they live. And by looking for patterns, researchers will learn more about what affects people's health. The National Library of Medicine and All of Us have prioritized citizen science as a focus for engagement activities with and through libraries and other community-based organizations to improve health literacy, digital literacy, and participation in scientific research. All of Us is a precision medicine study, so it's one way that researchers are looking to advance the ability to serve the public health needs of all people. It's a new way of looking at health treatment and prevention as being designed for individuals. So this way, doctors can take into account a person's lifestyle, environment, and biological makeup to figure out the best treatment for that individual. The neat thing is that we already use precision medicine in some ways, such as eyeglass prescriptions. So eye doctors can tell us exactly which lenses will work best for us, but when we visit our regular doctor, they might be more likely to prescribe the treatment or prevention that works well for most people, but everyone's needs are not the same. Next slide, please. So All of Us is building a large national database to help researchers eliminate one size fits all medicine, but they do need volunteers from all walks of life to advance this work. Next slide, please. So citizen scientists understand the importance of volunteering to advance scientific research, and the future of individualized medicine may depend on an army of 1 million volunteers, and scientists want you to get involved. So to learn more about All of Us and to learn more about participating, you can visit joinallofus.org/NLM. Thank you so much for listening, and I'll pass it back to you, Caroline.
Thank you, Tess. All right, next up we have Doosun Kim from the Eterna project. He's going to give you a brief introduction to the work he does and how you can get started with Eterna before we go into breakout rooms. Doosun?
Cool, yeah, thanks so much for having me, and thank you for being here, everyone. Please, if you have questions, feel free to put them in the Q&A box, and I'll try to answer them as I go on. If not, I'll get to the ones I don't get to answer at the end. I'm here to tell you about Eterna, which is a citizen science project that we started in collaboration with Carnegie Mellon about 10 years ago at Stanford University. I work in the DOS lab at Stanford University, which carries out experiments on RNA biochemistry. And Eterna is a really vital part of our research that helps engage citizen scientists to discovering design rules about this biomolecule RNA, which in recent times has come under quite a bit of public attention due to its role addressing the COVID-19 pandemic. And I'll sort of describe to you the projects that we have carried out in response to the COVID-19 pandemic in the last year. Invite all of you to join along as well. Caroline, do you mind going to the next slide? Thanks. So just as a quick walkthrough of how you get started on the Eterna, we've tried to lower the barrier of entry as much as possible. So you can just go to eternagame.org. You have to be over 13 years of age due to these regulations around video games. And you'll make an account, and it takes about 5 or so minutes. And then what we set up is a series of tutorial puzzles that are going to allow you to master the concept of Eterna through a series of relatively small, easy to solve puzzles. And you can see at the top there on the right side what they look like. They're named after machines in lab that you might frequently encounter as a aspiring scientist. And there's about 11 or so mini puzzles inside each lab that you will walk through. Overall, the tutorial sequence is about 10 labs in total. Like I mentioned, each lab has multiple puzzles, as you might see on the bottom left picture. And then after you complete the tutorial sequence and have really mastered the concepts that drive Eterna, we invite you to join us in the official Eterna labs. And the Eterna labs are puzzles where we take the solutions that are submitted to us by citizen scientists We actually synthesize the RNA molecules that you have designed on your web browser, and then we will synthesize them and test them in-house at Stanford in a laboratory with a professional scientist and give back to you the results of our experiments. And I'll describe to you the process in more detail as we go along. So just a quick aside, so I'm not just a totally random floating head. I'll describe to you how I ended up here. I did and hopefully my sort of diverse and unusual background will convince you that anyone and everyone can have an impact in citizen science. So in my undergraduate studies, I studied chemical engineering at the University of Texas at Austin. About 90% of my classmates went into the oil and gas field at the time. But I took a pivot and did research in the field called synthetic biology, which was just emerging when I was an undergraduate. I won't date myself, but it was some time ago where synthetic biology was a very, very new field. After that, I went to graduate school and got my PhD in chemical and biological engineering at Northwestern. I focused intensely on biology research here, especially in that field called synthetic biology. And then I happened to work on RNA machines in my graduate work. And through my work at Northwestern, working on RNA machines, I got in touch with a professor at Stanford that I collaborated with very intensely. And turns out that professor is Riju Das, who heads up the eterna project. And he would describe to me, oh, instead of just designing RNA machines at your desk by yourself, why don't you involve citizen scientists and allow people to design these RNA machines that you're looking to design? And I thought, at first, I thought it was a pretty crazy idea. And then after my PhD, I quickly joined him at Stanford, leading the citizen science project Eterna, and specifically leading our efforts in computational design of mRNA therapeutics, which I'll describe to you in the following slides. So before I go off to how Eterna specifically works on mRNA vaccines and the contributions that we're looking to make, I thought it'd be pertinent to go over the very basics of mRNA vaccines, why they're important, and how we tie in citizen scientists and video game players in helping design improved mRNA vaccines, in this case against COVID-19, but you can imagine we can then apply what we've done on Eterna to other infectious diseases like the flu and other many infectious diseases that are viral based. So the very basic concept is that a vaccine is a molecule that trains your immune system to recognize and fight viruses, and specifically infections arising from those viruses. So in the SARS-CoV-2 virus, which is the virus responsible for COVID-19, there are multiple proteins that lie on the outside of this virus. The E, which is short for envelope, the S protein, which is short for spike, and the M protein, which is short for membrane. Specifically, these proteins are what your body sees when the virus infects your cells. And I'll describe how mRNA vaccines utilize what the body would normally see to then train itself against infections from this virus, SARS-CoV-2. So in the case of COVID-19, I mentioned to you that these three proteins coat the outside of the virus. So the idea behind mRNA vaccines is, what if we, instead of the virus presenting to you the spike protein, your own cells will encode and present the spike protein of the SARS-CoV-2 coronavirus? Meaning, what if you had your immune cells express the spike protein and present that to you, to the rest of the immune cells, instead of the virus having to do it? So it looks like the virus on the outside, but it has none of the other bits that make you sick. So the vaccine encodes for this spike protein. And when I say the vaccine encoding for it, it's another more simplified way of saying an mRNA molecule that would encode for this protein. On the next slide, I have a diagram that shows you what processes in biology we are looking to take advantage of. So in biology, there's a central dogma of biology in which DNA gets converted into mRNA, and that mRNA gets converted into protein. And I told you that you could present the spike protein to your body and it would sort of help train your body against it. But to make that protein totally outside of your body can be difficult, and also many times is a very slow process. So as an example, the flu vaccine, which is also a very similar concept where you make a protein that is going to allow your body to train itself against the flu virus, takes about 6 months on average. So starting in March or so, the WHO releases which strains they'll use for the flu vaccine. An army of scientists and engineers get to making the flu vaccine, and by September, they will release the flu vaccine for that season. So you can see, it can be 6 months just to produce the right protein in vaccines. And mRNA vaccines seek to make this process much, much faster. So if you progress on the slide, I'll show you what process we are highlighting. So what we're aiming to do is, instead of making the entire protein outside of the cell and presenting that as a vaccine, scientists in the 1990s had the idea, what if you gave your body directly the mRNA to make the spike protein itself? Instead of the entire viral genome, which is what could make you sick, what about just the mRNA that'll make a spike protein? Give that to your body can then make the spike protein and sort of train itself against what that might look like in the case of an RNA virus infection. So we get to that, so that all sounds good and fun, right? You'll give the body an mRNA that will encode. Oh, sorry, I think there might be a switch in the, I want to wait to not make sure the transition is smooth. The mission that we're going for as a community of scientists is to design a better mRNA sequence that can code for the spike protein. And how we're going to do it at Eterna is, instead of relying on a bunch of scientists in a closed room, we like to open up the problem to anyone on the internet who has access to a web browser and also likes to play video games. And the reason this becomes a really tough problem to solve just through computer algorithms or just through exhaustive testing is that for the spike protein, in the example of the spike protein, there's 10 to the 600th power ways to code for the spike protein. And for comparison, that is just so many orders of magnitudes more than the number of stars in the Milky Way, and far more than the number of cells that exist in the single human body. So the amount of solution space that you have to search through is immense. So you can't do it exhaustively, and you need to brainstorm with many, many different people and gather ideas from every corner of inspiration that you can gather to sort of hone in on the solution as fast as possible. So we turn to citizen scientists because we on Eterna know that citizen scientists are amazing at sharing their ideas and also coming up with new ideas that we can test very rapidly in the laboratory. So this is where the platform Eterna, beyond what's on the web browser, comes in. So you can see on the top left, the player, goes and designs RNA molecules in a video game format, and then you can submit your solutions for a single lab or a puzzle. And what you see on the top right is other players who have played your puzzle will vote on designs based on shapes they like, based on the sequences they've taken liking to. Maybe they'll vote for your sequence because you're friends and they like you, so it pays to be very popular on Eterna. But we allow the players in a democratic way to vote for their favorite RNA designs. And then this is where Eterna really takes a turn, and we take the votes very seriously. So we take the top N, vote gatherer. So sometimes it'll be 10, sometimes it'll be 16. And we will synthesize the RNA molecules and test them using experiments involving RNA biochemistry at Stanford University. And then the unique part and the learning iterative part of Eterna, and why we have very good Eterna players, I think, is we will return the results of our experiments back to the Eterna players. So in turn, the scientists and researcher, which I'm part of that team, we help the players understand scientific data, how we're presenting it, and we work with players to better represent their data so that in future rounds they can learn faster. And a lot of times players end up bothering me for different analyses, and I learn a lot through their suggestions as well. And we go around and around this circle for a very complex problem in RNA design. And we found that within 5 or 6 rounds of this iterative design, testing, and feedback cycle, video game players can far outperform the state-of-the-art algorithms in RNA design. And just to show you what we've been able to achieve thus far on Eterna, on the top left, we are looking to solve a long-standing problem in RNA design. This problem in science called thermodynamically perfect ribose switches. This means that we are able to control the shape of RNA molecules from two to three different states with very, very precise switching using temperature or other chemical induced small molecules that may induce change. We are also tackling problems in rational three-dimensional design of RNA. This becomes very relevant in the human body. Very few things are 2D. You know, everything sort of exists in three-dimensional structure. So we're adapting Eterna to also have video game players think about what things look like in 3 dimensions. On the bottom right, we have an effort called Open CRISPR. CRISPR, as you might have heard, is a very popular technology. It won the Nobel Prize last year in chemistry. It is a gene editing technology that have been used in many, many different fields, from agriculture all the way to human health. And what we're helping to do in Eterna is help design this very important RNA component of CRISPR to make it much safer. So we believe that gene editing technologies have tremendous potential, but they are in need of advancements in abilities to control them so there's no runaway gene editing. So Open CRISPR is our effort to help do that. And then finally, on the bottom left, I'm showcasing some of our greatest hits of scientific publications. So these are papers and manuscripts that are peer-reviewed and published for professional scientists and researchers like myself to read in academic journals. And what's really unique about all the papers that I've shown here is that the authors, or the majority of authors, if not all of the authors, are actually Eterna video game players, many of whom don't have formal scientific training, and some who have no formal education beyond high school or very early college. So it really shows off what are video game players and citizen scientists, the level of science fluency that they've been able to achieve, and how they've really pushed forward this rather competitive and complex landscape of RNA design. So yeah, writing papers with citizen scientists is one of the most fun parts, because they just can't well, one, they can't believe how tedious it is, and it's great to share that part of doing science with them as well. But on the other side, it's just really refreshing to see, what's important in a manuscript. Like, what do citizen scientists believe are important in their findings to communicate to the community? And that often is different than what someone who does science as like a day job for me, like me, might focus on. So having that perspective of how you communicate the science back to a broader audience is really, really been transformative and helpful for me as a professional scientist as well. And then finally, I wanted to walk you through a quick demo of Eterna, what it might look like when you're actually playing the video game. So what I'm doing here is toggling back between the desired shape, which is an RNA molecule that's made to look like a pair of glasses, and its natural shape, what it looks like in this current state. So what I'm doing is changing some of these yellow or adenosine bases to blue uracil bases. So RNA is made up of 4 different molecules. We've included color coding so that if you don't want to learn the names of these 4 bases, you can just go purely on color. And we have players who are very advanced and work totally in color as well. And what I'm trying to do is figure out through iterative testing whether I can form these little pairs that you see. So you can learn that A and U form pairs and G and C form pairs, and how to achieve the shape of RNA that the puzzle lays out to do. So many Eterna puzzles are like this. Many are much more complex as the RNA molecule gets bigger and the shapes are more complex, but it's just a quick demo of what you might encounter on Eterna when you first start. Finally, but this is a super open invitation to come join us on Eterna. You can engage with us through all of our social media channels, but most importantly, we have right now ongoing our open vaccine campaign. The motto is that the vaccine that is going to be meant for everyone in society should be open for anyone in society to have input on and also help design if they would like. So you can find all of that at eternagame.org. And also if you are the generation that likes to do things on their phone, I'm too old for that, but I know a lot of people like to do everything on their phone. We've recently developed mobile apps that are deployed both on the Google Play Store and the Apple App Store, which we hope will also help increase accessibility to Eterna for people who maybe don't have ready access to a computer. So we're always looking for ideas of how to be more inclusive and include more people in the Eterna community. So if you have any suggestions, please don't hesitate to shout at us through any of our channels or just contact me directly with any suggestions. I have some questions I felt, maybe it was in the chat. Oh, I had a question from Michael who asked, in journals, is there any room left for the text if all the contributors are listed? So on some puzzles, we have up to, so for very complex puzzles, you're right, we'll probably have up to like in the hundreds of different players who submit designs. However, so what we do is we have an open Eterna players on the author list, and then in a supplementary table, maybe list all the players that contributed. But in the author list, we'll probably include just the players who contribute to the manuscript writing, who are often sort of the leads in leading the discussion in their community about the scientific takeaways of that puzzle. We have, I know historically, have had to have conversations with journals because some players identify strongly with their video game handle instead of their legal name. And in journals, there's no precedence of publishing under a video game handle. So we've had to go back and forth with publishers about how people can use their video game names to publish academic papers, which I think was a first time conversation for the editor as well. So everyone who has submitted a solution is cited in the manuscripts because that piece of data helped inform the science and the research, but not on the main author list because of space, like Michael pointed out.
I love that. The video game names, those can be anything, right? They could be like, I don't know, Silly Horse 77 or something like that. Oh my gosh.
They have to be like pretty PG and there's like a character limit, but yeah, people have just random, like there's a couple people who just random letters and numbers and you ask them and they're like, it's just random letters and numbers. So yeah, journals are more hesitant to publish a random string of letters and numbers as an author sometimes, turns out, but we made a solid case because we're pretty unique in the origin of the players and authors. So yeah, it was pretty fun.
Wow, yet another way Eterna is blazing a trail. Thank you so much, Too Soon, for getting us up to this point. Next up, we have Neha Joshi. So she is a senior at UCLA, and she's going to ask you a few questions before we go into breakout rooms. So as we discussed, everyone, this event's a little bit different from events we've done previously because we're actually going to stop the live stream and allow everybody to jump into another Zoom room to have full audio, full video, and be able to try Eterna out and ask you soon some questions. But so our folks on YouTube who are watching and folks who might be watching the recording after get the full Q&A experience, Neha agreed to join and ask you soon a few questions. So Neha, over to you.
Hi, everyone. I actually had questions about the open vaccine campaign. I was wondering if you could tell us a little bit about the progress that's been made in the last year or so that we've been in the pandemic.
Yeah, so maybe I'll elaborate on specifically the scientific problem we're looking to solve. RNA vaccines were the first ones to get emergency use authorization in the population, and they're the first ones to go out to people. And you might have seen on the news that RNA vaccines have to be kept very, very cold between the time they're made and the time they're given to people because they are known to degrade. Right, so you might think you're getting the full dose, but it's been sitting at room temperature for this long, so it's degraded somewhat. And we've known this about RNA molecules, that they degrade over time. So the scientific problem that we knew eterna would be very, very well suited to handle. Another thing. We know that RNA molecules degrade all the time. And we know that the more structured an RNA molecule is, meaning the different shapes that you have, we knew that the more interesting shapes they have, the less they're likely to degrade. So we had this mission of here's the mRNA molecule, here's the protein we need to code, and then let's make it as structured as possible so that they would be more stable in solution at higher temperatures than the existing mRNA vaccines. So that's the problem we went after. And we worked on it for now about eight or so months. And in the 8 or so months that we've been working on it, what we found were that the mRNA molecules designed by the video game players, again, outperformed the ones that were computational designed. And we were able to make mRNA molecules that were 2 to 3 times more stable at room temperatures and buffers than sort of the state-of-the-art starting sequence. So in the last year or so, we've been focusing intensely on making mRNA vaccines more stable. And we've shown that we've been able to achieve the jump that we think will be really essential for distributing the vaccine widely. And we're currently writing it up in a manuscript that will be shared openly with all access allowed.
Thank you. That's really, really fascinating. Thank you so much for sharing. My other question was about some of the regulatory hurdles that affect citizen science and maybe specifically the Eterna project or the open vaccine campaign. If you could elaborate just a little bit about that as well.
So there is no, so the regulatory framework around, so there's 2 sides of this. The first is the regulatory framework of doing science. So you have to, there are things like you have to be safe. You can't work on pathogenic molecules without the proper training. So there are the scientific problems that have regulatory sort of frameworks around it. And we have to make sure at Stanford, we stay within those frameworks. So if we're working with very, dangerous RNA molecules, we have to do those in certain rooms with certainly trained people. The other side of regulatory that we think about a lot is when you engage members of the broad public, it's not specifically outlined in legal text for what we know, but we think a lot about the ethics of citizen science. How do you make citizen science such that it is an inviting and collaborative effort with the scientists and not just sort of mechanical Turk style people just chugging along and using people for labor. So the way we try to address that on Eterna a lot of the times is to be very open in our community. And we also have a forum that coexists with the video games that people can go to the forums to make suggestions about the platform. They can go to discuss science and have a channel to talk to the scientists that are doing experiments. So we found that open lines of communication are probably our best bet to addressing the sort of the ethical concerns that you might have about exploiting citizen scientists as professional researchers. As far as regulations, there are things like if we were to take a citizen science design molecule, which is available and freely open. So one tenet of Eterna is that all the data we generate should be freely open and available to anyone. It's not particularly a regulatory hurdle, but if a large pharmaceutical company wanted to license that molecule, how do we make sure that the licensing process and the profits that are generated from that therapeutic can be equitably distributed and handled in the platform without, again, the citizen scientists being exploited for their ingenuity and work. So we have in the past and are continuing conversations with our interested corporate partners who develop medicines to talk about what the best way to compensate the community for their work might be, instead of them taking just a ton of profit and not giving any back.
Great. And it looks like we have maybe time for one more question before we go to breakout rooms. And thank you so much, Neha, for your questions. I see one in the chat. Michael wants to know, is this a problem well suited for genetic algorithms? And this is one thing I run into a lot with citizen science. I actually didn't know what an algorithm was until this year. I'm not ashamed to admit it. So when you answer that question, Dusan, if you could explain what a genetic algorithm is, and then address whether or not this is a problem well suited for it.
So genetic algorithms, if I understand correctly, are computer algorithms that optimize a lot like, they sort of try to mimic what happens in nature, meaning if you read into natural selection, there's this sort of survival of the fittest principle that governs a lot of the characteristics that you might find in plants or animals or other beings that have gone through evolution. So genetic algorithms refer to a broad class of algorithms that evaluate fitness of different metric, and then select ones with favorable characteristics, much like an evolving animal might be selected for over a long period of time in nature. So it is well suited, but the amount of compute you would need, because the number of possibilities that you would need to search through is very, very large. It often takes a massive, almost hard to fathom amount of compute to search through enough of the possibilities. I can share with you the paper on Eterna that is looking at the best folding algorithm was a Monte Carlo tree search algorithm. So in some ways it was a genetic algorithm, but not genetic algorithms alone, we have seen, I don't think can solve this problem, because there's a secret sauce of human creativity that we haven't been able to replicate via algorithmic searching alone. We've tried a bunch of different things like raising the temperature. So in machine learning or optimization, temperature refers to how widely how like wide of a net you're willing to cast on a particular problem. We've looked at different temperatures in our optimization algorithms, but so far we just haven't seen algorithms be able to outcompete the very elite video game players that we have on Eterna.
That is so cool. And I think for everyone listening, that's one really great thing about citizen science. You know, you don't have to know all these things up front as you participate, but you will end up learning really interesting things as you go along. So you'll be making a difference in the world while also enriching your own life. So some really cool things there. So before we end our live stream, we're going to go into a breakout room. This is our chance to give our attendees in the Zoom a little bit more of a voice. So for those of you who are watching on YouTube, your show is over. So Michael says, I think one can get a secret sauce at Walmart. Perhaps, but there really is a secret sauce of human participation. I think we see that again and again. So Dusan, before that we go to breakout rooms, is there anything else you want to tell our live stream audience?
Yeah, the biggest message I would say is to just go try it. I have found in the past, it's much easier to spend the 5 minutes to go try something and realize I don't like it, or if I do like it, it's great, than the 10 minutes I would spend otherwise deliberating if whether I would like something or not. I think the thing about Eterna is it really draws you in. So if it sounds interesting to you, we've lowered the barrier as much as possible for getting started. So I would really encourage you to go check it out on eternagame.org, and then join the community. So Eterna is much more, when you join, hopefully, and you do, it's much more than you just playing video games against the screen. It's really about the conversations that you'll have with the players on Eterna. We have a chat box that's scrolling on the right side that you can always tune in on, and people are talking about the most random things, but mostly RNA. And there's a Discord and a forum that people communicate in. And so it's a really big community of not only video game players, but citizen scientists who don't like to play the games, but like to be involved in the discussions, the professional researchers at Stanford, and also our collaborators at Northwestern. So it's just a wonderful community of people. So yeah, I would love to see as many people join in as possible, and hopefully talk to you through Eterna very soon.
I love that. That's awesome. And if you all come up with a fun username on Eterna, that could appear in a scientific paper someday. So absolutely great. And all of these resources we talked about today are available on scistarter.org/NLM. And that can get you to the Eterna website as well. So I'm going to go ahead and stop the live stream now because we're going to go into our breakout room. So goodbye, our friends on YouTube. We hope to see you at the next event, and we hope to see you on Eterna sometime soon.